So
hello.
See and hear you. Awesome.
We're good? Wonderful. So hey everyone, welcome to our presentation concerning the design and synthesis of our new rivastigmine analogs. So starting off, we're just going to give you guys a brief overview of neurodegenerative diseases. These diseases affect millions of Americans every year. Over 5 million suffer from Alzheimer's and almost about 1 million suffer from Parkinson's. Neurodegenerative diseases essentially happen when the cells in the nervous system don't work the way that they're essentially supposed to. And then they generally get worse over time, essentially why they're called degenerative diseases. This degeneration shows why it's important for us to start treating these diseases as early as possible to prevent some symptoms such as memory loss, anxiety, or depression. So unfortunately, there aren't any actual full cures that can completely eradicate all of these diseases. However, there are medications that have the capacity to mitigate symptoms, especially in those with milder cases. In simple terms, these medications help by boosting the receptors they can see on the bottom of this diagram. Essentially, what happens is these receptors receive acetylcholine, which is the chemical that more or less allows nervous cells to communicate with one another. And then neurodegenerative diseases either decrease the amount of acetylcholine or they inhibit the receptors. And then the receptors is what acetylcholine binds to after getting released. And this is more or less what allows nervous cells to communicate with one another. So this is a visual for just about how we come up with these analogs. So we start from the natural product, which is it's just our nature part. And then this is Physostigmine. This becomes a synthesized version to increase potency and also increased ability to get manufactured. And then we improve on these rivastigmine with our analogs, which we create through synthesis. And once these analogs are properly synthesized and we confirm them with NMR, we can test the potency of, which is essentially how well it can inhibit cholinesterase. And then we do this through bioassays. So a little bit more about Physostigmine. This was found in the calabar bean, which is pictured to the left, which was first discovered in the 1840s by Europeans, even though locals had been using it for what's called a trial by ordeal, which is pretty much when anyone who is standing trial is forced to eat these beans, which are, by the way, very toxic. And then those who can finish and survive are deemed innocent. Further research was done throughout the 17th and 18th centuries before Otto Loewy discovered the effects of Physostigmine on the parasympathetic nervous system, which led to the creation of Physostigmine as treatments. And onto rivastigmine. Rivastigmine is the FDA-approved cognition-enhancing drug commonly used for the treatment of diseases such as Parkinson's and Alzheimer's. The synthesis of rivastigmine is essentially used by allowing the nerve cells to communicate with one another much better.
So regarding the mechanisms behind how physostigmine works in the brain, it is essentially a competitive mimic of acetylcholine. And what this means is that instead of acetylcholine binding to the enzyme acetylcholinesterase, we see physostigmine binding there instead. And so just for clarification, acetylcholine is a neurotransmitter that is highly involved in memory, and acetylcholinesterase is the enzyme that breaks down it. And so when there's less acetylcholine, the memory is going to work worse. And so essentially, for the mechanism of physostigmine binding to acetylcholinesterase, there's an active site nucleophile, which is serine 200, depicted in the red. And there's also an anionic site on the acetylcholinesterase that the respective parts of acetylcholine and physostigmine will bind to. And so because physostigmine essentially takes the place of acetylcholine, it binds to acetylcholinesterase in the same manner. And so here you can see that the acetate that is highlighted in yellow will bind to the hydroxylate group, and the nitrogen that is highlighted in blue will bind to the anionic site. So physostigmine isn't the ideal agent, however, because number one, it has a very poor bioavailability. Number two, it has a high toxicity. And also, we've seen that there have been very difficult synthesis of this product, and there's also been challenges with its isolation. In fact, we saw that some chemists have found that there's only been 15% yield of physostigmine in a 10-step reaction, in 10 steps, and 3 steps had to use pyrophoric material. So again, this is a very dangerous synthesis. And so the mechanisms of rivastigmine are similar to those of physostigmine, except they're better in some aspects because rivastigmine is much less toxic, and it also has a better bioavailability.
Okay, so on the screen here is a current basic overview of previous synthetic efforts. So on the screen is what we call a synthesis of S-rivastigmine. So on the arrow, the compound to the left is called 3,1-dimethylphenoethylenol, which is one of our reagents. And then through a one-step acetylation, it forms the rivastigmine compound on the right. And there have been previous synthetic efforts to synthesize this molecule to achieve better selectivity. Some previous efforts we used to optimize the reaction are shown on the screen above. So to optimize the synthesis of S-rivastigmine, we tested out 2 different factors. So one is solvents, and then one is the base or the catalyst. So our solvents included BCM, and then our bases include pyridine, triethylamine, and DMAP. And as you can see, our reaction with BCM and DMAP and pyridine had the best yield of 57.3%. So that's the one we decided to use. So synthetic efforts are also shown on the screen above. So again, the 3A one, which is on the top, that's the S-rivastigmine. And then 3B is something we call S-dimethyl, and then 3C is something we call S-diethyl. And then finally, 3D is called S-morph. And what difference mostly in these analogs is the carbamoyl chloride. So for example, for S-dimethyl, we use something called dimethyl carbamyl chloride. And then for S-diethyl, it's called diethyl carbamyl chloride. And you can guess what the other ones might be. But according to our experiments, S-morph had the best yield out of all of these analogs. So we want to see, once we make these analogs, if they actually work. So if you remember, Spencer mentioned how our study revolves around something called acetylcholine, which is this neurotransmitter that's important in the body. And this enzyme called acetylcholinesterase inhibits it to form something called choline. So our analogs are supposed to bind to this enzyme and inhibit it from functioning. So by measuring the amount of choline we have, we can honestly measure how effective the inhibition is, because the more choline there is, the less effective the analog is inhibiting. And then again, the less choline there is, the more effective the analog was. And this is a slide about our S-enantiopure analogs. So we had 2 main ones. It was S-rivastigmine and S-rivastigmine and R-rivastigmine. And what difference in them is that the structure, and we decided to focus on S-rivastigmine analogs because they're more potent, and we wanted to see if that logic held true to all of the other S analogs, such as S-diethyl and S-morph. And overall, S-ones are just more effective than the R analogs. And finally, we want to thank ASDRP and all of Children Foundation, and also our advisor Ed, and then all of our group members for all of their help, because we wouldn't have been able to do any of this without them.